Thank you everyone for coming tonight. As many of you know, we're in the process of rebooting our in-person community lecture series, so I really appreciate having everyone turn out. This is something that we started all the way back in, I want to say 2017, maybe 2018. 2017. Yeah, and was really a ton of fun before we hit the pandemic. We had built up to an audience of high school students and outside community members, I was just telling Maren here, of something like 100 to 150 people once a month, which was really, really fun to be involved in. So I hope that we're going to have that coming back again. It's really tough to do these hybrid, so we're trying to stick with it in person for a while. We'll see how it goes. My name is Eileen Anderson. I am the current director of the Sewinville Growth Stem Cell Research Center. This is, I think, a part of our mission at UCI. So UCI really has three elements in terms of UCI Health that we think about: discover, teach, and heal. We do all of those things within the confines of this building in the Stem Cell Research Center. Discover, of course, is our basic science research program, which has many different facets, and you can easily go to our YouTube channel and hear more about the different sorts of research that's going on here. Heal is kind of an obvious one, being situated in the College of Health Sciences. Of course, this is about translating what we find, what we discover in the lab, into human clinical trials and then hopefully into clinical practice. We have a number of videos that are from these presentations that touch on that topic as well, because we have some really stellar translational research programs that are within the confines of this building on spinal cord injury, my own work, traumatic brain injury, on cancer, on skin disorders and baldness, how to use stem cells to cure baldness. We have a new initiative that's all about cell and tissue engineering and using tools, the tools that stem cells present to develop the cures and therapies of tomorrow. But tonight, in part, what we're talking about is that third arm of our mission at UCI, which is about teach. And a really critical element of what we do, and in fact what California Institute of Regenerative Medicine, CIRM, and the National Institutes of Health Fund is really about teaching. Undergraduate level teaching is kind of obvious. That's what a university is. It's what we're built around. But actually there's all different kinds of elements of teaching that happen within an academic system, and you're going to get some flavor of that tonight from Peter's presentation. Certainly the California Institute for Regenerative Medicine has some really key programs that are based around this idea of teaching at the undergraduate level, at the graduate level, so PhD students, at the clinical level, MDs who are looking to do research or learn about clinical research, and postgraduate or postdoctoral positions. And this is our workforce in California for regenerative medicine therapies. So that aspect of workforce development and the academic teaching mission is something that's really, really critical and important. In that regard, I'm going to give a quick little shout out right now, which she's not anticipating. Monique Williams is at the back of the room. Dr. Williams, can you stand up and give a wave? She's like, what? So for our community lecture folks, Monique has Dr. Williams has just joined us from UC Riverside as our academic coordinator for the center. We're building out a pillar of education here. That's really, really important because we have so many different aspects of training now. Brian Cummings, who's here in the front of the room, directs, which he and Monique are frantically working on this week, an undergraduate training program called the CIRM Compass program that it will almost fully fund 5 undergraduate students a year who are in need in terms of completing their education and interested in working in the STEM field for everything about their tuition and fees and additional set of training programs so that they can leave UCI and enter the California workforce in the area of regenerative medicine, like trained in a way that couldn't happen anywhere else. So these guys are just kicking off that program this summer. We also have a number of graduate training grants. Peter is involved in many of them. I'll introduce him in a moment. A CIRM Scholars program that is for PhD students, for postdoctoral students, and also some clinical fellows. A P30, that's a National Institutes of Health training grant that focuses on skin cell biology and stem cells, which we also host here in the Stem Cell Research Center. A neurotranslational and stem cell biology training grant that's funded by the National Institutes of Health, NINDS, Neurological Institute, what is that? Neurological Institute is in the thing. Oh my God, I had a long day today. NINDS, which Peter is also one of the directors for and has been really, really important in terms of our graduate training here that we can offer at UCI. So all of these training initiatives together mean that we really need to build out this additional pillar. So thank heavens for Monique, who has joined us, and for all of our faculty who really have taken on the extra workload that comes with offering these training opportunities. With that, and my training spiel, I'd like to introduce Dr. Peter Donovan. Please rise. Rise. Peter, there, see. Peter is the founding director of the Sue and Bill Gross Stem Cell Research Center, and without him, we would not have the building that we're sitting in tonight. And so that's why the people who are in the know gave a nice round of applause for him. We're delighted to have him still be such an integral part of our center. And training is really one of his passions and something that he has just continued to take on a huge leadership role for us. I think I'll leave it there because I went a little long. And I get all excited about the training. And I'll turn it over to Peter to continue with his slides. And then Dr. Donovan is going to introduce our second speaker, Dr. Pein.
Thank you so much, Eileen. I always tell people that one of the reasons why this is such a great building is that not only was it designed well, but there's a group of people in it, including Eileen and Brian and the other faculty, who we all get along well and help each other, and I think that's why it works so well. I thought I'd begin by telling you a story about myself many years ago when I was growing up in London. And my brother and I one night decided we'd go to the pub, and we were going to our local pub, and somewhat unusually, we took my father along. And by the way, that's what British people do, they go to pubs. Americans tend to go to baseball or the movies, British people go to pubs. And we took my father, which was a bit unusual because he didn't drink that much. He would occasionally have a glass of wine with Sunday lunch, or if they had, friends come over one evening, he might have a scotch and soda. And my mother never drank, so he didn't really go to pubs, but for some reason we took him. And so we went to the pub, and my brother and I had a nice pint of beer, and my dad ordered a half of some weak lager, like Coors Light or something, And it was a really unusual night. This was our local pub. We went there all the time, and for my brother and I, there was no one there that night that we knew. Nobody at all. And yet every 5 minutes, somebody came up to my father and said, "Hello, Mr. Donovan. Lovely to see you. Haven't seen you in ages. How you doing? And, great to see you. Hope you're doing well. Thanks for all the help you gave me." And so my brother and I sat in the corner with no one to talk to except each other. And my dad was mobbed by all these people. And so you might think, well, maybe he was a secret alcoholic that went to the pub all the time and just didn't tell us. But it turns out my dad was a teacher. And so all the people that came up to him that night were students he taught many years ago and recognized him and came to say hello. And my father wasn't even a secondary school, high school teacher. He taught elementary school and middle school. So these were kids that 10 years after they left his school, at least, they came and said hello to him and thanked him for what he'd helped them with. And I think that has given me a perspective about teaching that I saw again recently at a high school event where there were 100 kids on in the band, and one of the teachers had taught 90% of them over her 27-year career. And teachers give so much to children. They give them their knowledge, they teach them how to learn the knowledge of other people, and they give them their experience as well. And what do they get? Nothing in return. Teachers are not paid very well, for one thing, but they get nothing in return except the joy of seeing those kids go off into the world and do well. And over the years as a scientist, I've had my 5 minutes of fame, in the spotlight, where I traveled the world and met interesting people and ate their food. And I realized, though, that no matter what I could accomplish as a scientist, and I'm very proud of what I accomplished, no matter what I accomplished as a scientist, the people that I trained were able to achieve much, much more. And I'll tell you why in the next couple of slides before I introduce Maren. So this slide is from 2009. It was a press release by UCI, which announced one of our first training grants from the California Institute of Regenerative Medicine, CIRM. And it was a picture of myself.
Who's that young-looking guy on the left?
Yeah, I know, right? Yeah. The camera adds a lot of weight. It turns out time adds a lot of weight too. Myself and Ping Wang, who's a colleague here, head of our diabetes center, and some of the trainees in our training program funded by the California Institute of Regenerative Medicine. And don't forget, this was at the outset of the pluripotent stem cell field, and California was right at the forefront of helping students learn and come into this field so that we could expand the field here in California. So I think there are 9 trainees in that picture, and I'll tell you a little bit about each one of them. And I only did this afternoon, and it's kind of amazing. Raymond Wong was a postdoc in my lab. He now works at the University of Melbourne in Australia and leads a lab working on pluripotent stem cells to develop new treatments for macular degeneration and retinitis pigmentosa. Yiting Lin is a medical research specialist at a college in the East Coast, I think, called Harvard. I've heard of it vaguely. And she continues to develop technologies for imaging cells and tissues in patients and for using radiotherapy to treat people with various types of cancer. Sung Hoon Ha works for GE Healthcare. He, again, uses imaging technology, and he's a RF engineer that develops imaging technology to image aspects of the brain and tumors so that we can see where different cells are and detect disease. Duncan Liu works for a local company down in San Diego, having moved at some point to Becton Dickinson, where he was head of their GMP products. He now works with this company down in San Diego, and he's risen to the rank of senior vice president. Christina Freuder, she was a postdoc on the grant. She's now at the University of Copenhagen in Denmark, and she leads the research group working on Alzheimer's disease and frontotemporal dementia, again using pluripotent stem cells to model and to develop new treatments for those diseases, technologies that she learned here at UCI. Hal Nguyen, he apparently is a real estate tycoon, and I see him as a potential donor. I really do. I think that might be his role. Sharon Rossi was a graduate student here in the lab of Hans Kierstad. If you remember, Hans was here. She worked on spinal cord injury, and now she works for the Bright Focus Foundation, who develop funding models to help support researchers doing work on Alzheimer's disease, macular degeneration, and other debilitating diseases. Desiree Salazar, who was a student in the lab of Eileen, she now is a major player at the National Institutes of Health, and Desiree's role really helps kind of steer the nation's funding in the area of cell biology, which is an incredible role to have and one that's very important. And then finally, Helen Fong, who was a graduate student in my lab, works at Sangamo up in the Bay Area, where she uses stem cell technology and genetic engineering approaches to develop new ways of studying Alzheimer's disease and other neurodegenerative diseases, including Parkinson's, again using technologies that she learned here at UCI and then developed further at the Gladstone Institutes and at Sangamo. So one of the incredible things about this slide when I went to look at it is just how successful these kids have been. And although Ping and myself were lucky enough to get the funding to support these people through the California Institute of Regenerative Medicine, it was also, of course, their faculty advisors that helped them to mature as scientists, to learn from their experience and then go on into the world and be successful. So for me, I've come to the realization that it's not what I do, it's what I can help other people do in terms of going out to do science. Now, these are all incredible people, but we kept two of the best for last, and we kept them here at UCI. Two other postdocs that were funded on the training grant. One was Munjal Acharya, who's now an associate professor here at UCI. He works on the effect of radiation on stem cells in the brain and how it, when people are treated with chemotherapeutics or radiation to treat tumors, how that can affect their memory in the brain. And he has a lot of funding from the National Institutes of Health, the National Aeronautics and Space Administration, because NASA is very concerned about what happens to astronauts in space, and from the California Institute of Regenerative Medicine, to understand how radiation affects stem cells and how it might affect the treatment of various diseases, including glioblastoma. And Matthew Bletton-Jones, on the right, was a postdoc here in the laboratory of our colleague Frank LaFurla, who's the Dean of the School of Biological Sciences. He is now a very well-established Alzheimer's researcher. He studies Alzheimer's frontotemporal dementia, again using iPS models to make cell types in the brain to really develop some of the first avenues of research into looking at cells in the brain other than neurons. So both of these young scientists who trained here as postdocs, we were lucky enough to keep them here, and they're doing a fantastic job. They're both at the forefronts of their field, and we're very lucky to keep them. So this was my point at the beginning, that we as scientists, we might make our individual discoveries, but really I think our impact is how we touch the lives of individuals, and they in turn touch the lives of other people. Matthew and Munjal now have trained a lot of young scientists in their lab, and they're out in the world also affecting the way that we look at various diseases and how we develop new treatments for them. So this top-down approach allows us to really have a massive impact, in my opinion. And when we renewed this program recently, we got some very nice accolades from the reviewers who just told us what a great program we have running here. And as you'll hear when Maren talks in a minute, I think we've continued to really recruit some outstanding students, postdoctoral fellows and clinical fellows to the program. And I think that some of the things we do here at UCI really are at the forefront of training young people, and we need to always be thinking about new ways to do that. And that's one of the great things about the colleagues I have here in the Stem Cell Center. But I'd like to end by saying how much your support matters. The renewal of the CIRM funding by the state of California was very important, in my opinion. In the initial funding effort, as you may know, it was put on the ballot because the federal government was going to ban all research on pluripotent stem cells. And so California stepped up to the plate and, in my opinion, saved the stem cell field at a critical point in its development. And the reinvestment by the people of California has meant, really, that that initial investment has been supported and expanded, and it's why, in my opinion, California leads the world in stem cell research, because of the investment of the people of California and the support through their taxes. And so I'd argue to you that there are a number of ways that individuals can continue to support this effort. One is by advocacy, when you have the opportunity to tell your representatives the things that you support, continue to support this agency because it has not only a dramatic impact potentially in the long run on the health of individuals, but frankly on the industry of California. The biotech industry in California is the strongest in the world, and this type of investment really helps that. Secondly, through philanthropy, we have these grants to support young people, but they don't support everything they do. They don't necessarily support students' ability to go to travel and learn new techniques, or travel and present their work and be critiqued. And over the years, we've had fantastic support from donors, and those donors have given small amounts of money that we've been able to really multiply, so that they give us, $1,000, and then we use that to do a pilot study that allows us to go and get a grant for maybe $20,000, and then that, builds on that to get even more funding. So even a small amount of support really matters. And then the final thing you can do is volunteer. We have a number of events at the center where members of the public come. For example, the Huntington Disease Foundation at UCI really supports research here. They have fantastic symposia where members of the public come and learn about that type of research, but also people come and support the events. Recently they had a team that ran in the OC Marathon, and you can probably tell I wasn't one of them. So I'd just like to end, before I introduce Maren, by saying training young scientists, in my opinion, magnifies our impact in this world. Because if Maren now goes on and trains 4 or 5 students, and they go on to train 4 or 5 students, the magnification is immense. And in my opinion, humanity has never faced a greater problem or set of problems than it does now. And so we really need these young people to go into the world, and we need young people to come into science and continue to be interested in science and continue to do science, because we face enormous challenges, and we really need these young people right now. So one of those young people is Maren Pein, who's sitting here. Maren received her doctorate from the German Cancer Research Laboratory, which is based in Heidelberg, Germany. And then she was recruited here to the laboratory of a colleague of mine here, Kai Kessenbrock. And we're delighted to have you, Maren, and we'll look forward to hearing about your work. Yeah.
All right. Thank you very much, Peter, for introducing me. And I would also like to thank everyone who joined today for this evening lecture. And I would like to spend the next 15 minutes or so to tell you why and how we're trying to build a reference atlas of the normal human breast with so-called single-cell and spatial resolution. And I will explain in a few more minutes what that actually means. So you may already know that breast cancer is unfortunately a really, really common disease nowadays. It actually affects 1 out of every 8 women in the US. Moreover, breast cancer can be deadly, especially when it's diagnosed in an advanced stage. And in general, the earlier breast cancer is diagnosed, the better are the chances of treatment success and eventually survival. So in order to understand what goes wrong in breast cancer, we really need to first understand what the normal healthy breast looks like, so that we could be able to detect the earliest changes that lead to tumor development in the breast. So what do we know about the human breast? The female breast is attached to the chest wall, of course, over different muscle groups. And each breast contains between 15 to 20 of so-called lobes that are further substructured into smaller units called lobules. And lobules are the milk-producing functional units of the breast. They look like these tiny bulbs, as you can see in this schematic, made of epithelium, and they are connected to each other via thin tubes called ducts that converge into larger ducts that transport the milk and eventually reach the nipple. Now, when a physician or a pathologist looks at breast tissue under the microscope in a so-called H&E image, as you can see here, they can distinguish different structures of the human breast, including the lobules that I just mentioned, the ducts, fatty tissue or adipose tissue, and fibrous connective tissue. And zooming into one of these lobes a little bit more, you can see in this H&E staining here the epithelium that makes up these lobules and these ducts. And it's these lobular and ductal epithelial cells that have the risk of eventually developing into breast cancer. Now, throughout the whole ductal and lobular system of the breast, the epithelium of the breast is composed of two cell layers. So there's an inner cell layer of so-called luminal epithelial cells, and these luminal epithelial cells are surrounded by a single layer of so-called basal epithelial cells. And this two-layered epithelium is further in contact with other cells that are non-epithelial, which can also talk to these epithelial cells and influence them and their behavior. Now, as I already mentioned briefly in the beginning, we are trying to build a reference atlas of the human breast to understand what the normal composition of the breast is in health with all the cell types. So therefore, we have two different goals that complement each other. The first one is to record all the individual cell types that are present in the normal breast. And in more scientific language, that means we want to achieve so-called single cell level. And the second goal is to create a map that allows us to know exactly where every cell of the human breast is located within the tissue and who their neighbors are, so which cells are talking to each other. And in more scientific terms, this means we want to have a spatially resolved atlas. Now, how can we make sure to build a really representative and reliable reference atlas of the breast? Well, in order to be a good atlas, the atlas should be two things: representative and comprehensive. So in order to be representative, we need to make sure that we capture many tissues from many different individuals, from women of different ages, different ethnicities, body shapes, etc. And we achieved that by collecting a lot of tissues from many different women. So far we have over 125 different women who have donated tissues to our research. The other important thing, and I think Peter also mentioned that a little bit, the importance of team spirit in these scientific endeavors. So this, in order to achieve a really representative atlas, we really need to have a lot of researchers and clinicians work together to achieve this goal. And we have over 40 people, researchers and clinicians who work together both at UCI and at different places in the US to achieve our goal. And the third aspect is that we want to analyze all individual cells that are present in the human breast. And to this end, we use a technology that is termed single-cell RNA sequencing. How that works is that we obtain breast tissue from the clinic after surgery, and we first dissociate this tissue, which means that we detach all the single individual cells from one another so that we end up with single cells from which we can then extract RNA and sequence that RNA. And by sequencing this RNA, we gain information about the genes that are active in the genome of every individual cell, so that we can then cluster the cells into groups of cells with a similar gene expression pattern or gene activity level. And this lets us define what kind of cell types we have in the human breast. Now, the other aspect of a good atlas is that it's comprehensive. And in order to be comprehensive, we need to have a representation of the entire organ of the breast. So we need to really analyze all the different parts of the breast. So to achieve that, for a number of different individuals, we actually were able to collect whole mastectomies. And what that means is that the entire breast sample was removed in a surgery and donated for research. This, of course, much more rare, and happens, for example, in individuals who have cancer in one breast, and then the other breast is not affected, but they decide to remove that breast in order to prevent that there could be cancer developing in that breast later on. We then work together with clinicians to come up with a strategy to define specific sample regions that cover the entire breast. So we collect simultaneously at least 16 different tissue pieces per breast that cover all different areas of the breast, from nipple to all the different areas that we have. And finally, to understand where the cells are located within the tissue, we combine our single-cell RNA sequencing technology with so-called spatial methods. These spatial methods allow us to place very thin sections or slices of breast tissue onto glass slides, and then stain these tissues with different molecules to detect RNA or protein inside the cells and inside the tissues, and get a really nice look at what the tissue looks like inside the body. Now I'm going to show you some results. So here you can see an overview of our single-cell RNA sequencing data. And what you can see in this nice overview is actually over 700,000 cells from more than 125 individuals. And in this representation, every cell is represented as a single dot. And in general, the closer together two cells or two dots are, the more similar their gene activity is. And you can also see this here in this heat map, that there are groups of genes that have very high activity, indicated in red, in particular groups of cells. And this allows us to define a cell type of the breast and to distinguish them from each other. So here you can see them labeled in this representation. And looking into only here epithelial cells of the human breast, you may remember from my introduction that there are two cell layers of epithelium in the human breast, but now we actually found that there are three different cell types that make up the epithelium in the breast. So the luminal epithelial cells, or the inner layers of epithelial cells, are subgrouped into two different cell types. One is called secretory luminal cell, or LUM-sec cell, and one is called luminal hormone responsive cells, or LUM-HR cell. And we didn't stop there, but we could even zoom in further into the individual epithelial cell types, for example, these luminal secretory cells, and further subgroup them into smaller groups of cells with more particular gene activity levels. And we could find that they are very diverse, with more than 7, or with actually 7 subgroups in the luminal secretory cells. We did this for all the different cell groups that we found. And here I'm just summarizing in this diagram that we found 12 cell types in the human breast, and they are composed of 58 different cell states. Now, one thing that was very interesting to us was that we actually found some cells in the luminal epithelial cell group that didn't only show characteristics typical for luminal epithelial cells, but also characteristics of basal epithelial cells that are normally around these cells. So they are almost like an intermediate cell type or cell state. And so what we wanted to do with that is to find out, can we see these cells inside the tissue? And to look at that, we used the technology that is so-called spatial technology, where we stain tissue sections. So here you can see a very thin tissue section of the breast with antibodies to detect proteins that are typical for luminal or basal epithelial cells. So I will zoom in here. So luminal epithelial cells are marked by an antibody with a green color. and the basal cells are marked with a protein that detects basal cells and is marked by red color. And what you can see indeed is that there are some cells that have both green and red staining, so they actually have this intermediate appearance of having both luminal and basal cell characteristics. We also interestingly found that these cells are not randomly distributed, but they seem to be more common in ductal areas of the breast. As you can see here, more yellow than in lobular regions. And even more intriguingly, we found that this intermediate luminal-like basal cell phenotype is linked to be more, or is more frequently found in breasts of women who have a very high risk of developing breast cancer. So these are women who have a mutation in the so-called BRCA1 gene, and this gene mutation predisposes these women to a very high risk of developing breast cancer in their lifetime, which is over 60%. And we found that these basal-like luminal cells are expanded or more frequently found in breasts of these women as compared to breasts of women who don't have this gene mutation. And finally, we also discovered using another spatial method that there are many immune cells in the human breast, and they are actually very close to epithelial cells. So here you can see cell nuclei of breast tissue cells, and a structure that is highlighted here is a duct, and then there are some lobular units as well. And in this tissue section, we actually stained for many different RNA molecules this time, which represent different cell types. And in green, you can see RNA molecules that are typically found in T cells, which are immune cells or defense cells of our body. And when you overlay and highlight the cell boundaries of the individual cell types, you can really see that T cells are found within the ductal structures and the lobular structures of the breast and very close to the epithelial cells, both basal cells in blue and luminal cells in orange and red. So there's a potential that these cells actually communicate with each other. Now, I would like to summarize. In order to understand breast cancer better, we need to first understand at a good level what the healthy breast looks like. And in order to do that, we're building a reference atlas of the normal human breast. This should have different characteristics. The first one is that it is at a single cell level, which means that we record all individual cells and analyze them using single cell RNA sequencing technologies. The second one is that the atlas is spatially resolved, which means that we can visualize where every cell that we identify is located within the breast. The third aspect is that it's representative, which we achieved by using tissues from many different individuals. And the fourth one is that it's comprehensive, which means that we have representation of all different tissue areas within the breast. We have made different discoveries that I introduced to you. For example, we found that there's 12 different cell types in the human breast with almost 60 different cell states. Within the epithelium, we found that there are some cells that exhibit characteristics of both luminal and basal epithelial cells, showing an intermediate characteristic of these two different cell types, and they are linked to developing to a high risk of developing breast cancer. And finally, we also found that there are many immune cells in the breast, and especially T cells are in very close contact to epithelial cells, which wasn't known beforehand. And I would like to just briefly thank people involved in this study. As I mentioned, there are many, many more that I can even put on this slide. Especially I would like to highlight my supervisor, Kai Kessenbrock, and co-mentor, Devin Lawson, my lab members, clinical collaborators, all the patients, the CERM training grant. I'm very thankful to receive funding through this and be part of this exciting program, the core facilities, and our outside collaborators as well. And yeah, I'm happy to discuss more and open for your questions. Thank you.
Actually, maybe before we start, because I just think it's interesting to contextualize, can you go all the way back to the slide where you show the H&E stain?
Mm-hmm.
In the room. So this is a hematoxylin stain, right? Straight H&E stain. Anybody know where hematoxylin comes from? Yes, that's true. So anybody, seriously? No. What is horseshoe crab? That's C1Q. So hematoxylin comes from a tree that in the Yucatan. And when explorers started exploring South America, like in the 1500s, 1600s, it was identified, I don't know why, it must have been some like indigenous people thing, but it became a dye that was used in the fabric industry in Europe. And those dyes that were used in the fabric industry in Europe are where most of our early histological stains came from. Because prior to having things like hematoxylin, there are a few other examples. As we started to study the body and understand how different that organs are made up of cells and where cells are localized, we needed to be able to stain those and identify what was what. And hematoxylin is one of the very early dyes that was used for that in the late 1800s and early 1900s. And then as you can see from Maren's talk, how much our technology has changed in the last 100 years, because now we don't just look at hematoxylin and say, hey, there's a few different structures that are there. You can identify almost 60 cell subtypes by virtue of breaking that down with how much technology is advanced. I just think that's cool. So I thought I would point that out. Questions from the audience?
Great presentation, by the way. Oh, microphone, sorry. I was going to say thank you for that presentation. I have a family history of breast cancer myself, so I think this is really important material. I know you talked about the clinical prevalence of breast cancer as well as the intermediate luminal cells like showing a predisposition. I was just curious if you could maybe tell me more about like the clinical implication of having this map, if that's going to be more at the detection level or the treatment level or both, if you could just discuss that a bit.
Yeah, no, sure. Yeah, I think that's a really important question definitely to ask. I think our research is a little bit more towards discovery. So I try to highlight that a little bit. We need to, I think early detection is definitely very, very good. You can see that statistics show that it's just really crucial to catch cancer as early as possible. But yeah, we still don't even understand what normal looks like. So the part or the aim of this this reference atlas of a healthy human breast is more to establish a baseline and a range of what normal looks like, so that we can see very early on, for example, in women who have these gene mutations in their germline, we can already see without them having any tumor that there are some changes that may predispose them or that we can monitor and then watch. It's not going to be a very quick translation into treatment. I don't think it's more discovery-based. But yeah, the idea is that we use that, and we will be able to use that to map out whatever can change. And these intermediate cells are just one example of what we found. Like, okay, there's a change, that we wouldn't be able to tell if we didn't have this reference of the normal, healthy human breast. Sure.
Thank you. Of the 40-some people that you showed, are they working on prevention or are they working on treatment?
It's very different. They are like a very diverse group of people who also, many of them are clinicians, so they work in the clinic, they treat patients. We have pathologists, we have people who work in other projects that are working in treatment too. So it's a really, I can't even tell you, like, it's only this or that. It's super diverse. People have their different background, their different skills, and there's all of it, I would say.
But is there a plan for treatment or prevention versus treatment? Is it split that way?
I wouldn't say so. So, for example, even in our labs, we have projects, and even the same people, may work on a project focused more on prevention and another project that's focused on treatment. I think it's really crucial to have both arms pushed forward. There's not one thing that is better or worse. I think we really need both, and we need both of them to improve. So this of course is more an example of how we could potentially improve the prevention or the early detection, but that's by far not everything that we're doing or our labs are doing.
Hi, thank you for
Hi.
your presentation. So I just had a question about the transition, like epithelial cells. So you said that they have like basal and epithelial like characteristics. I was just wondering like if they were more like, are they similar to stem cells in the way that they can transition from like one cell type to another cell type? Like, do they have like that sort of phenotype, which is pretty persistent with cancer, or is it more like their own thing?
Mm-hmm. That's a really, really good question. So, well thought, well thought of for sure. It's actually, that's the interesting thing. Like we see now, they are there. They are somewhat in between, but where do they actually come from? It looks like, based on what we can tell so far, that potentially they come rather from luminal epithelial cells that then go backwards in their kind of differentiation, which means that they can lose some of their cell type commitment, and so to say, become more progenitor-like. But we don't, I guess it's very challenging to prove that. And to actually track that, especially within humans, like we can't just model something. But yeah, that's a really interesting question that we're trying to figure out right now. But yeah, the idea is, or the current hypothesis, I would say, is that these are cells that have lost their lineage fidelity, so they kind of degenerate towards a more progenitor-like phenotype.
Oh, hi. That was a phenomenal talk. And I had a curiosity question as far as you guys building this atlas. Do you plan on putting it in some kind of database, or is it, okay, that was my question right
Yes,
there.
yes.
And will you guys be
Great
contribute,
question.
like, is it gonna be something that multiple labs can contribute? Because I know breast cancer is a really huge thing. And, it's phenomenal. You guys had such a large, like, sample size of, every demographic that you guys can think of, because diversity is very important when it comes to, this kind of research. And there are some, ethnic groups that are disproportionately, diagnosed with breast cancer and they're not given the proper care. So the fact that you guys did include that is great. And I was just curious on how you guys were going to distribute that. So it's good to hear it's going into a database. That's great.
Yeah, no, great question. And that's definitely something that also still needs to be improved even more. Right now, I think we have mostly maybe like over 40% Caucasian and 41% African American, I think, and then some Asian, but we still need more representation for sure. To the question about the database, yes, everything is publicly available, or will be. We have a bioarchive right now, but this study just got accepted for publication and in Nature, and it will be available very soon. And there's actually even going to be a website with all that data. I also wanted to mention it's actually part of a really big consortium. So this is like many different labs working on this, and it's funded by the Chan Zuckerberg Foundation, which I don't know if you've heard it, but it's like this human cell atlas project, which aims to really understand all the cell types in the human body. And they really also push for open science and posting all the different data, like the raw data that you have. So we actually will have everything, all the raw data available for the scientific community and whoever wants to look at it. Yeah.
Fantastic. Do you have one? You have to wait, Cameron. Someone really needs to pester Peter soon.
No, we can leave Peter alone. I wanted to ask, what are your career aspirations? So based on this training that you've had here, what do you intend to do next?
I mean, we will have to see. Right now, I'm a postdoctoral fellow, so for those of you inside academia, you know, like there are certain milestones you want to achieve and have to achieve to publish your papers, secure some funding. I definitely like science very much, so I'm very interested in pursuing this further, and maybe starting my own lab if possible. But yeah, we will have to be open and see where life leads me. But I definitely am still driven to do science for sure.
Hey, great talk. I was just curious about what criteria went into getting the patient samples, if any, what might have bias mitigation might have been done, or if you think that the total donated samples is enough to overcome that kind of bias.
Yeah, that's a great question too. So we tried to be more inclusive, of course, for screening like all the different patients. So we have people who undergo so-called reduction mammoplasty. So these are women who have very large breasts and suffer from them due to just the weight of it and back pain, etc. So this is one source of our samples where just like tissue pieces are taken. Then the other source is cancer patients who have also a non-affected breast. So they, of course, that is already like not a fully healthy patient maybe, but the breast tissue per se. And the third one is prophylactic mastectomies, or samples where people know they have a family history of breast cancer, for example, and therefore they either for diagnostic reasons or preventative reasons donate their tissue. For other criteria, I mean, the main thing was pathologically normal tissue, so no cancer. I'm not sure. I think we tried to span a lot of different ages, of course, from adulthood to older ages. No current treatment most of the times, but we even have some, because the reason for that being, if you undergo, for example, chemotherapy, a lot of the cells are basically wiped out and may not be fully treatment naive, so not fully normal, let's say. But yeah, that's a great question for sure.
Maren, thank you for that great talk. I wondered if I could ask a precursor to Brian's question. You got your PhD from the German Cancer Research Foundation, which is, I think, the largest research institution in Germany and a well-renowned cancer research place. Why did you come to UCI? What was it? I don't mean that in a funny way, but
What possessed you?
no. You probably had lots of opportunities to go all over the world, perhaps. And I'm curious, what was it about UCI that you thought, yeah, I want to go there for the next stage in my career?
Yeah, good question. No, definitely the driving force was the science. I think it was a time, so I was just finishing up my thesis, and me and my partner were looking for opportunities where to go next. I definitely knew already I wanted to go abroad. I wanted to explore a little bit more, go to the US. And then I had followed like the scientific publications that are coming out, and there was a paper by Kessenbrock, who's my group leader now, whose lab I joined, that I noticed, and I found it really beautiful, and that kind of intrigued me. So I put in his name and Googled, and thought, like, where is he? Oh, UCI, what is that? Like, checked around. Then I found, together with my partner, also Devin Lawson's lab. They are, for those of you who may not know, they are both professors here at UCI, and they're married. And Kai's lab is focused a little bit more on the earlier stages of breast cancer, and Devin's lab is more focused on the advanced metastatic stages. And that really combined perfectly what I was interested in and what also my partner is interested in. So we just kind of watched them like do their thing. There was a time, I think, where they secured some funding, and we thought, okay, that's our time. Like let's shoot out some application, and yeah, then it just worked well. We came here, interviewed, and yeah, of course there were different options as well, but it's just the whole package. I loved also the community here. I find UCI is really collaborative and curious. That's like what science should be for me. So the whole package just fit.
It was a great presentation. I was wondering if your research also focuses on the imaging technology, and I also wondered if you think that the imaging technology is a limiting factor for creating a comprehensive atlas for human cells.
Yeah, great question. Definitely, we have. So I don't know how much background you have in different imaging technologies, but yeah, there are definitely very different technologies that are better at doing one thing, but they have other limitations in other areas. So what we need to do is for every question that we have, to get to the right answer, we need to find the technology that fits best, that helps us answer that question at best. So we're actually using different technologies based on the questions. We let the questions kind of drive what we need. We let the questions direct us into the technologies that we need to answer these questions. And then if we don't have that expertise in that technology, we need to find people who have that expertise, and they can do it for us, they can help us do it, they can teach us. That's normally how we do it. Hope that answers your question.
Yeah. Yeah, good talk. And just follow the previous question. So you talk about it's a good idea to build a database for the normal breast, the cell type, but the source you get is from, basically it's kind of like not completely normal. So I just curious, can you get something from someone died from the accident or something? Have you tried that? Or
yeah, I mean, reduction mammoplasties are basically normal. There's no there's no other reason, or these individuals don't really have anything else other than they may suffer from the weight of their breasts. So otherwise they are healthy. But there are also sources where women can just donate tiny biopsies of tissues. I mean, no one who is entirely healthy and has no medical reason, I think, just goes into the clinic and says, here, I want to give some of my tissue to you. So it's a little bit more challenging, but there are databases where people actually donate biopsies, at least. So that's definitely something that can be used, and that in this current cohort hasn't been used by us yet. But other groups also have done that. I think that came up earlier too, that of course many people are looking to collaborate and to build on this data set. But that's a good question for sure. With diseased, did you mean that, like diseased autopsy samples? It may be a little bit more difficult to get consent. I mean, you have to definitely be careful with like what or how you define normal. On the other hand, it's also a question like who is really normal, like who is healthy 100% at every time. But yeah, autopsy samples would be great to have. It's just very challenging to have the okay, because we can't just take tissues from someone, right? Especially with technologies like sequencing, there's concern about data being able to track the data to the person. So those samples are a little bit difficult. And then the other aspect is that we often need fresh tissue samples, so we can't just have tissues that were, like, if someone dies unexpectedly by an accident, you can't just rush in there, get the sample, and then bring it to a lab. And it just logistically doesn't work so well. So, yeah, we have to work with what we have and what we can get. But it's definitely a very good point. Thank you
And
for
these
that.
are such a sensitive set of techniques that
Yeah.
you have to have really clean samples,
Yes.
no delay between
Exactly.
death and autopsy.
Yeah.
I enjoyed your presentation immensely, especially as a young person in science who has had plenty of mentors myself. I hope to also take the way they've impacted my life and pay it forward. With that being said, in my limited experience of like tutoring, mentoring, teaching in a certain way myself, I've also learned that the learning and the teaching is often reciprocal sometimes, and we like learn from our students. I was just curious if you wanted to share maybe a lesson that stands out that you learned from a student, or just speak on the reciprocal nature that can come with teaching.
Yeah, I think that's a really, really important point. And one of the things that I am troubled about in universities, and I think it's in academic research in general, is bullying, where because of a power differential, a faculty member kind of bullies the people under them. And to be honest with you, graduate students can bully undergraduates too. And one of the reasons why I think that's so dangerous is that that means that one person's intellectual idea drives everything. And one of the great things about having students in your lab is that they bring ideas to the table as well. And an incredible thing about scientific training is that you can start off working in one lab where you learn, let's say, fly genetics, and then you move to another lab where you're maybe working on cancer, and then you move to another lab where you're working on genomics. So it's created a unique scientist who has those three different aspects of study. And I think that's the beauty in a way of science. And so when I think of labs, I want the students to bring their own ideas to the table. I'll tell you two little stories. I took an idea to my postdoc mentor, and it was a stupid idea. And he said, let's just forget we ever had this conversation. And then, so I'll never tell you what that was.
And that idea was?
No. I ain't gonna ever repeat that. And then I had a student in my lab, Helen Fong, who is now at Sangamo doing, genetic engineering on cells to model various neurodegenerative diseases and building systems to do cell transplantation. And I remember very much towards the end of her PhD, she would argue with me about stuff. She said, I don't think it's all about this gene. I think there's other genes. And I would say, no. And I realized that she had gotten to the point where it was time for her to leave. Not because she was arguing with me, but it was beautiful that she felt confident enough then to argue. And honestly, that's what you want in a lab. You want people to come to you and say, no, I don't think that's right. It's much more important for students or postdocs to disagree with you than agree with you in a way.
I disagree.
So yes, I think it's absolutely learned a lot from everyone. And not just in terms of science really, about how to do things and how to approach different. I had a technician who, it was when I first started my lab, we were dissecting a tiny little piece out of an embryo, and I was really good at it, just saying. And then within about a week, she was doing it faster than I was. And I thought, and then she'd figured out a better way to do it, so it's all good. Yeah, much better that way.
That's all the nature of the exchange. And
yeah.
so Peter, maybe we can close on this question. What role do training grants and training opportunities play in that, right? That technology transfer and developments and the kind of progression that we make from like hematoxylin staining to doing spatial transcriptomics.
Well, I didn't know hematoxylin came from a tree in the... Where did eosin come from?
The root.
There you go.
You're lying, aren't you? Training grants. Usually when students or postdocs come to university, they will work in the lab of a PI, a faculty member, on a particular topic. Like Maren works on, defining cell types in the breast. And they're kind of fixed in that world. And I think what training grants bring is they bring an ability to train people in other things that are absolutely essential for success in science. And I actually give UCI a lot of credit in this realm. I think UCI leads the country in this type of thing, in part because of structures that were set up on the campus. And the California Institute of Regenerative Medicine also played an important role, because they said, if we're going to fund you, you need to be willing to go and talk to the public and explain your science, and work with patient advocacy groups to explain to them what you're doing, why you're doing it, and why we're giving you money to do it. And so that pushed us to really think outside of the box. And so we began to engage people to teach students and trainees to speak well in public, to communicate with a lay audience, to discuss things in a way that patient advocates would understand with a sensitivity, and also to learn the other soft skills that are so required for success in science. When you're running a lab, you're basically managing people, and everyone is different. And we've done training to try and figure out, how you work with difficult people, how you deal with difficult conversations, how you can use different people in a lab to enhance the success of the group. Brian and I were talking about this the other day. He loves Excel, loves it, loves it, loves it, loves it. I hate Excel. I'm not even sure I know what it does. But I realized at some point, we were doing a training thing together, that I needed people like him who knew how to do Excel. I could do the big picture stuff and talk to an audience, but I needed, colleagues like Brian to get everything really well organized. And then you realize that you need lots of other colleagues who have other strengths. And so we both realized that, and those are the types of things that are value added that the training grant brings, that unfortunately individual faculty members who are paying for someone in their lab maybe think, no, I just want them working at the lab bench. So that, I think, is one of the great things about the training grants. And I have to say, I was really pleased that our CERM training grant was ranked #1 in the state. Because also the NIH training grant we wrote in stem cells and neurodegenerative disease, it's almost become a poster child for the National Institutes of Neurological Disease and Stroke. And I think we do it really well, and so it builds our reputation, and hopefully when a young scientist like Maren is thinking about where to come, she will see that and think, yeah, that's a place I like to be.
So that's a perfect spot. Thank you both so much, and thanks to our audience.